My name's Caroline Dean. I'm a project leader at the John Innes Centre in Norwich, which is a very big research institute looking at fundamentals of plant and microbial science. My seminar today is going to be about how plants sense and remember winter, and I've got into that through studying the molecular control of timing of the transition to flowering. Well, it's a real pleasure to be here. I really envy you guys because, as Osselin says, there was nothing like this when we were coming through as undergraduates. And, you kind of have a time to sort of network and meet people, but also talk about a whole range of things that might just start an idea in your mind. And I put this picture up because this was the sort of idea that occurred, it wasn't even an idea, it was a sort of, I noticed it. And actually we had breakfast this morning. I forget who my colleague was chatting breakfast. When we were looking at the beech tree outside, there was this beautiful copper beech. And we was like, well, why are the leaves purple? So we ended up in a conversation about anthocyanins and how they are the sort of sunscreen for the leaves and how good anthocyanins are for you. And that set up a whole series of questions, I think, which actually could go on to be research careers. So you might find sometime during this week, a little spark sets up a kind of kernel of a question in your mind that actually you may 30 years later be still working on, because that's what happened to me. So I put this picture of a cherry tree because when I was growing up, we had a series of cherry trees outside my back garden, and I used to love spring. Isn't it? Everybody loves spring. Flowers come out, everything feels better. But it was always like, well, why do they all flower at the same time, all this row of cherry trees? And actually, if you look in a neighbourhood, all of the same trees of the same variety will, within a couple of days of each other, flower at the same time. Now, not all plant species flower at the same time. They have staggered flowering, but within the same variety, they will flower very similar to each other. So why is that, So when I had the opportunity to set up a lab, I thought, yes, I want to know why it is that plants flower when they do. Now, at the time, and it was 30 years ago, we knew quite a lot about flowering physiologically. Gardeners how many people like their gardens? There you see, we've got a reason. The plants in the garden, some need long days to flower, some need short days to flower, the chrysanthemums. Some, if you go and visit the desert, they need water to flower. There's a whole range of different physiological cues that makes plant flower. So I guess the presumption at that point was there's going to be lots of different kinds of molecular mechanisms behind this flowering control. But it's important to learn and important to study. So we need to start somewhere. And so that complexity was perfect for this Arabidopsis system. Because Arabidopsis, the idea is it's a very small genome, but it's also a very easy plant to do genetics on. I imagine you'll see some this week. A lot of you have heard of it already. But the idea is if you want to study a process, you just make a mutant in that process, in Arabidopsis, because it was easy to get those mutants, and then clone the gene out and see where it takes you in terms of the biology. So that was what we started, right at the beginning, no prior work. We just said, we want to study flowering time. Another colleague of mine, George Coupland, who started at the same time, was doing flowering time as well. And there was a couple of colleagues, one in Holland and one in Spain, and we got a little group together. And we worked very productively for 5 years, lots of EU funding. And actually, the other thing in science is that you get to be very good friends with these people. You see them year in, year out at conferences, and they get to be your buddies. Anyway, what happened over probably the first 5 years is we sort of got a skeleton of understanding of the genetic pathways regulating flowering time in Arabidopsis. Now, I'll show you lots of details, but I thought I'd start off with some sort of schematics that make it easy to understand. And remember, at the time, flowering looked really complicated because every type of plant needed a different cue to flower. And yet what emerged out of the genetics is very simple. You take the Arabidopsis, you do a mutagenesis, generally with a chemical. You then look for that rare individual that does something different from all of its siblings and then study it. It's a mutant in a gene required for that process. And so we did lots and lots of genetics. And what emerged was that actually there's lots of inputs in Arabidopsis that affect flowering. The temperature affects it, hormones, day length, light quality, various temperatures when it's growing. But all of these things converge to regulate a common set of genes. And actually that was the clue, because then that common set of genes, when actually you look at all these genomes now, and because genome sequences are coming out every week, there's another big genome sequence, these sets of genes are in all of these different genomes. So when they are sort of activated to a sufficient level, the plants will undergo the transition to flowering. And in fact, what became clear was that different plants then use these different inputs differently. So in fact, this big complexity of flowering didn't actually have to be really difficult to understand. It's just that through evolution, some plants particularly use day length, and so that pathway was the predominant one. Others, and I'll tell you about, generally needed to flower after winter. So this was the predominant one. But because they all fed into this, activating the same set of genes, you could then explain and understand basically flowering in all organisms. And certainly that's the simple hypothesis that actually still we haven't really maximized and exploited in terms of trying to breed and manipulate flowering time in a whole range of plants. So, you know, people think, oh, flowering, Caroline's done it for 30 years, so's George. It's all sorted, all done and dusted. But actually, you know, most of these things up here, we still don't really understand how they all input. So big area, big potential. Now, so the other little story, and again, it's an absolute true story. So we had all of this understanding, and that helped us really understand how these environmental endogenous inputs triggered a set of genes, which then caused the plant to flower. These basically caused the meristem, allow the meristem to switch from making leaves to flowers. So the question is, now what am I going to focus on? Because there's a whole wide range of things here. And what particularly intrigued me was this requirement for winter. So I grew up in the northwest in Manchester, and so seasons were just one of those things you got used to. Fairly sharp seasons, fairly obvious. And then I went and was a postdoc in California. And actually, they have seasons there, but, you know, they're much more discrete, sort of, you know, winter's nothing like winter in Manchester. And I thought, oh, it's, you know, again, it sort of occurred to me without really being a specific question. And then I went to a shop and I bought some tulip bulbs because I was feeling a bit homesick and I wanted some tulips on my balcony. And the chap said to me, put them in the fridge for 6 weeks. I was like, wow, this is a bit weird. You know, you actually put the bulbs in the fridge for 6 weeks before you plant them. And actually that one comment has made such a huge difference in my life, because it's like, I actually went up and looked in the textbook, and like, why do I need to put them in the fridge, you know? And this process called vernalisation, which is what I'll tell you a lot about, was why you needed to put them in the fridge. They actually needed the prolonged cold of winter before they would flower. So it's not that, you know, I know, as a teenager, I imagine, well, plants don't do much in the winter because it's cold. Nothing much happens in the cold. Then it warms up and the day lengths get longer, then they do something. That was the sort of assumption. But no, they actually need the cold. And so that's what we've focused on. So you say to yourself, well, how do plants know they've had winter? And again, you think, well, you know, Caroline, you make a big deal of this, but surely it's just monitoring cold for a certain amount of time. And that can't be that hard, can it? Well, so when you then look at what a plant experiences, so we're doing some field experiments, which I'll briefly allude to at the end, and we have some sensors in the field at the level of the plant, okay? And we've monitored them over the last 4 years. So from July, this is actually in northern Sweden, but you know, this is the daily fluctuation in temperature that a plant is seeing through you know, the end of summer, beginning of autumn. Now, there's a nice kind of constant bit here because there was 3 foot of snow on the ground, so the sensors were nicely protected. And then, boy, you can tell when spring is, the snow's melted and boom. So the plant's having to work out what is going on in this massively complex fluctuating temperature regime. Okay, so, you know, winter actually isn't as straightforward as you think it's going to be. Okay, so now we work on Arabidopsis. I thought I'd better show you some Arabidopsis. Probably, this is the Arabidopsis that you imagine. This is the one most people work on. It's the rapid cycling type. And of course, you work on the rapid cycling type because you want to do rapid genetics. And probably most people in the world would do the rapid cycling type because it goes from seed to seed in about 6 weeks. So it's really quite quick. But actually, if you go outside here and collect Arabidopsis seed, or anywhere further north than here, and then you put them in, you germinate them and you put them in your nice long day chamber, about 70% of what you would pick would actually grow like this. They grow quite happily, but they just keep making these. They seem to have forgotten to go into sort of that early rapid flowering mode. They're called winter annuals, and actually from about here northwards, this is the most common reproductive strategy. We can debate why that might be. So anyway, you have these two flavours of Arabidopsis flowering, winter annual and rapid cycling or summer annual. And this is the same seed. This is the same genotype. And this just shows how totally responsive to the environment the flowering is. So it's very plastic, but clearly the plant has to get it right, because if it flowers at the wrong time, in terms of producing seed fitness, which is really what organisms are really worried about, it's absolutely disastrous. So massive controls on flowering time, because it's got to get it right. It's got to read the environment and work out when is the end of winter, when is it okay to flower in the nice conditions of spring, because, you know, you've got all your very tender bits hanging out in spring to flower, so it needs to be a nice condition to actually maximise reproductive success. And so that's the question that we wanted to get at. So I started, 30 years ago with the same three questions, and my middle name, everybody laughs at me, is Mrs. Focus. It's like, God, Caroline's just so focused on all this stuff. But actually it served me well over many years. And so, I could have given a seminar 28 years ago saying, this is what I'm about to start doing, and here it is, the same three questions. But was really, why do some plants adopt this winter annual habit and some have rapid cycling? So why do they do that, and what's the molecular basis of that? If you do overwinter, how on earth do you register winter as a plant? How do you figure out these fluctuating temperatures and know that winter's over? And of course, winter's not the same thing in different parts of the world. So how is Arabidopsis adapted to different winters? Now, the genetics, and I'm not going to go through an awful lot of the genetics. There's lots on the website, which is in your booklet. There's all of the papers and all the summaries, and if you want any more information, it's all there. But I just want to reinforce the importance of this, as Ottoline said, for agriculture. So I'm talking about Arabidopsis, and you might say, well, does it really matter that, an overwintering Arabidopsis, and you can tell me what the mechanism is? But if you then look at our crops, our important crops, they are also winter annual and summer annual varieties for wheat, for all the barley and rye, for brassicas. And actually, the ability to breed winter and spring varieties in our major crops has made a huge difference to the geographical range in which they're grown. So I show here, we've got oilseed rape. Most oilseed rape in this country is winter sown, but sometimes further north in Scotland it's spring varieties. Again, the spring varieties of cereals, very important indeed. And actually, as we go through, Arabidopsis is a very close relative of the brassicas. And what I'm going to tell you, we've actually got a parallel program. It's exactly the same in all the brassicas as it is in Arabidopsis. And actually, that's going to have a pretty big effect on breeding brassicas in the next 10 years, I think. The cereals are probably slightly different target genes, but all of the concepts of these multiple inputs regulating, combining and integrating, again, is all very similar. So we actually have some very good conceptual understanding now to really be able to manipulate and breed varieties which are going to be more robust to this inevitable climate change. Okay, so we've got our winter and spring types of Arabidopsis, just to reinforce, which is the same as winter wheat and spring wheat. Same genotype, treated differently, look very different indeed. And through lots of genetics, why is some winter annual, some spring? Why does it register? We've done it all through mutagenesis. We could have ended up looking at probably 50 different genes, but no. All our genetics kept coming back to one target, which was very useful as far as focused science is concerned. But this target, think of as a brake to flowering. If you express this protein, the plant stays vegetative. So it basically is a transcriptional repressor that keeps off all the genes that are required to switch the meristem to flowering. So if you think of it as a floral brake, a repressor, its name is flowering Locus C, FLC, which is a terrible name. It was named before we kind of discovered what it might be doing. And sometimes I think we should just change the name to make it something easy to remember, but everyone says, no, Caroline, you can't do that. But anyway, FLC. That's the name of the most of my talk will be about. So as the seedling germinates, if this is made, then you basically get that phenotype where you just keep making leaves. Okay, the meristem is blind to long days, warm temperatures. It cannot flower, it won't flower early until you've got rid of FLC by prolonged cold. And what happens during the cold is that you shut off the expression of the gene, and as I'll show you, you keep it off. So this is the really important player in Arabidopsis and the Brassicaceae need for winter. So I'm going to talk about this vernalisation, this need for winter, the ability to sense and remember winter, my title. What's very interesting is that actually the cold releases the brake very slowly. Now remember, the plant's got to not get confused by a short day or couple of days in autumn and think, haha, I've had winter already, it's okay now, I can switch FLC off and flower. So what it does is it switches it off very slowly indeed to make sure it's been through the whole of winter before the brake is completely released. And this quantitativeness has intrigued people for many years. And in fact, if you take the plants out after 2 weeks cold, this is what's called a northern blot, where you're looking at RNA levels of this particular gene. No one ever does it with RNA blots these days, but hell, it's an old slide. Anyway, it's just very nicely, easy to see. So you've got lots and lots of it with no cold, and less of it's made as you give as the plants increased cold exposure. But if you take them out at 2 weeks and then keep the plant growing, it stays at that 2-week level, sort of intermediate level through the rest of development. And it flowers a bit earlier, but not as early as it could do. If you go at the full 4 weeks, now it's completely switched off, now it flowers very rapidly. So it's a quantitative response because of quantitative silencing of this gene by cold. And this memory of wherever you got to is what's this epigenetic process that Ottilie referred to. Now I'm going to put this onto a schematic through a life cycle of a plant, and I'm going to build on this schematic as I go through. So here's our gene, schematically, and this means here, this arrow means the gene is being expressed, the RNA is made, the protein's made, keeps off all the genes required to switch the meristem to a floral fate. Now the plant, so this is autumn, now the plant's beginning to feel the cold and actually slowly, as I showed you, switches off FLC. And now spring comes and actually it stays off. And it's important it stays off. The brake is off, so the plant can now respond to long days, to warmer temperatures, probably all the endogenous cues, and that's then triggering flowering. So all of these multifactorial things are important, but only when this brake is switched off. The plants then flower. But interestingly, at some point, and we now know it's as the seed are developing, this is a little seed pod here from the plant, the gene is switched back on again. Because, of course, every generation of plants you want to make sure needs to vernalize. So you've got to switch it off, but then switch it back on again. And so we're going to, first of all, talk about what we know about this epigenetic bit, the remembering the cold exposure bit. And of course the plants are growing very rapidly at this point, but it keeps the gene off. And then the second half of my talk, I'll go back to what we currently understand about sensing the winter, because that's the bit we still don't really know the mechanism of. Okay, remembering winter. So as I said, you start off with Arabidopsis. This is the beauty of it. You don't need to know anything about what the underlying mechanism is. You generate mutants, and then you see where those mutants take you. So we started with a line, one of our lines that needed vernalisation. We treated it with various mutagens, vernalised it, and most of the population flowered early. But actually 1 or 2 of them didn't, they flowered late. So this contained a mutation in a gene required to promote flowering in response to cold. So what I wanted to say was actually I'd really stick your hand up if I say something that doesn't make sense, because much better to follow as I'm going through than later. So be brave if I say something you don't understand. Anyway, so when we eventually cloned these things out, and this required, we didn't have a genome sequence at that point. Can you imagine? You can't just dial into a computer. We didn't have insertion mutants. Nothing like that was available. We had to do sort of fairly basic chromosome walking, generating libraries, map-based cloning. That's all now in place. It's very easy to clone a gene now. But anyway, when we did it, it took us into the world, which I'm going to tell you a lot about chromatin silencing and epigenetics. Specifically, a chromatin silencing process called polycomb, which had been actually developed, identified in flies in Drosophila. So I'll tell you about polycomb. And then the other area it took us into were two proteins. It doesn't really matter what they are, but these are very important in vernalisation, Vin3 and Vern5. And one of them was only expressed in the cold. So that was a kind of nice, if you like, link to the physiology. Okay, so we get into polycomb. Now, as Ottling says, who's heard of Arabidopsis? And there was a good response. Who could explain to me, or who's heard the term epigenetics? So maybe about half. So this is really, really important term, and it's something you'll see in the newspapers lots and lots. So it's actually very important, and what the understanding that's coming out of plants is actually really helping us understand the importance of epigenetics. So if we've got drawn here now the chromosome, and you unravel your chromosome, you'll see that the DNA, which is completely unraveled here where the base is contained in the chromosome, wrapped around these, what are called, if you think of it as a necklace, these are the beads of proteins, histone octamers. So histone octamers, 4 times 2 different pairs, make up what's called a nucleosome. So think of it as a bead. The DNA wraps around that bead and then is compacted down to make up the chromosome. And to make a gene expressed, you need to, in the cell needs to unwrap it, allow it to be expressed, and then pack it up again and put it away, because there's so much in each nucleus that you can't open everything at the same time. So this is why it becomes very important for gene regulation. It's not only these memory processes, but chromatin has huge effects on whether genes can be transcribed and how they're regulated. And I heard this, and I thought it was potentially quite useful. You can think of this in terms of a computer analogy, which you may be more familiar with. So the DNA, I should have missed one thing off here. So you've got the DNA wrapped around the nucleosomes, and the DNA itself can be modified chemically, a methylation group's added, or in fact, little tails of the necklace, the histone tails, these little squirrely bits here, they can also be post-translationally modified with chemical modifications. And those two things influence whether or not you're going to be able to unwrap or expose the DNA for all of the enzymes to transcribe it. So this analogy with the computer analogy is you can think of the DNA and the protein as the hardware. You kind of need it, but it doesn't give the right instructions. In a computer, you then need to bring along a set of instructions with software to instruct what's going on and to deliver these various chemical modifications to tell those bits of the chromosome, those genes, whether or not to unwrap or not, whether or not to stay closed. So, that may help. It's an analogy people use quite a lot for the epigenetic versus the genetic regulation of the genome. Now, this is where it becomes important. So I'm going to tell you about one particular kind of regulation. There's lots of different epigenetic regulation. But when we look at it in terms of much more broader biology, it turns out a lot of the differentiation from your stem cells as you're an embryo to the different cell types in our own bodies, this is all maintained by epigenetic modifications. Because, of course, every cell has the same DNA, but every cell turns into something different. So all the different genes are transcribed differently. A set of instructions is delivered in the embryo, but often those instructions go away. And then that cell, in a certain configuration of which genes are being made, through many divisions, remembers those instructions through many cell divisions. That's this epigenetic regulation through, generally, modifications of either the DNA or the beads. So I paused here because people get a bit confused about epigenetics, but it's not actually very complicated, but it's such an important concept. It would be worth now, if it's confusing to anyone, stick your hand up and say, hang on a minute, what do you mean by that?
Is it always methylation that happens? Is it always methylation? Or is it...
No, I've said methylation on the histones. There are acetylation, there's ubiquitination, there's phosphorylation, there's and probably a whole range we don't even know about yet. On the DNA, it's mainly just DNA methylation. There's different flavors of DNA methylation. I think that's still true. But, you know, I think we still have a long way to go to fully understand all this. But anyways, subtle chemical changes. Okay, so it's important for development in our own bodies. Twins. Have you ever thought about, I mean, genetically identical twins aren't actually genetically, you know, phenotypically the same. So lots of diseases that come out of twin studies have an epigenetic basis. And there's quite a lot of interest now, arthritis, schizophrenia out of twin studies, which are clearly epigenetic differences. And actually now, as we've got lots and lots of cancer genome work going on, lots of funded work in the UK, a lot of genomes seem to be correlated with, whether it's causative is still a bit of an issue, but with changed epigenetic status of very important genes are involved in many cancers. The BRCA1 is often methylated, the colorectal ones is a different gene. So, you know, this whole how genes are regulated, how they're switched on when they should be, how they're switched off normally, when that goes wrong through the epigenetic misregulation, that's when cancers occur. So you'll be hearing a lot about epigenetics in the next few years. And this is the other one I put in because my graduate students said, oh, it's so cute, you got to put that one in, everyone loves looking pussycats. Anyway, the classic case of epigenetic regulation is in females. We have 2 X chromosomes, okay? One of those has to be shut down to get the dosage of your X right, so that you have the same amount of X expression as your male brother. And so there's a very famous X inactivation which has been studied for probably 50 years or more. And the calico cat shows it beautifully. So in our bodies, a female, so one of the X from my mother, one of the X from my father, which will be slightly different from each other, randomly, in different cells in the embryo, one of those is shut down. And so that's sort of drawn here. So this bar body, which actually you can see on a microscope, it's shut down and then compacted into a little knob. And so people studied this cytogenetically many years ago. One X is shut down, but it's random. So, you know, in one cell, it might be one of your X's, in another cell, it's a different one. And it happens early enough in the development of the embryo that on the calico cat, you have on one X an orange coat color, and on the other X you have black. And so as these then develop in these clonal regions, what you have here is a representation of, in this patch, one of the cat's X's had shut down, and then all of the cells from that are regenerated from it, and that's why you have this patchy calico cat. It's a mosaic. Now you can see that on a cat, but all the women in the audience, we're as mosaic as that, we just don't actually see it, okay? It's an interesting thought. But the thing that's actually I want to stress, and actually as we come to some recent stuff, is it shows that the epigenetic regulators, and I'll tell you about the various proteins that do this, but they don't necessarily work on all the copies of the gene in the same cell. That, you know, if you read a textbook about transcription, you say, oh, this transcription factor's switched on and it goes off and finds all its targets. That's a trans network of regulation. In fact, epigenetics is very much a cis-regulated mechanism, because here, you know, all the proteins are the same in this cell, except those 2 copies of these alleles, or the different chromosomes, are reacting differently. So there's something in the local chromatin that is distinguishing those 2, in this case, X chromosomes, but in other cases, polycomb targets. So a cis mechanism is an important concept too, and the X demonstrates that beautifully. Okay, so let's get back to winter before I completely run out of time. So we cloned these genes out. It took us into the world of polycomb, and now we've understood what we mean by epigenetics. What the polycombs do is that they modify the tails of the proteins. And in fact, they add on a modification, which is methylation in this case, onto histone 3, the lysine at position 27. So if you read these textbooks, they have these sort of names, H3, H4, K9. Anyway, it means, you know, which histone, which position, and this is trimethylation. And there's a series of proteins that basically add that onto the histone 3. So we're going to color code them. These are for off. So they're color coded red as a traffic light system. So on the nucleosome, you're going to have your off modification. And one of the things we discovered, I'm not going to get much into, is that where you always have an off, you have an on. Because anything that's stably maintained, if you're ever going to be able to change it, you have to have something balancing it in a sort of opposition. And this happens to be the modification for on in our case. So we have our gene, FLC, on in autumn. We've got the various modifications on, the chromatin's open, the gene's exposed, the gene can be transcribed, making the protein, and that's what keeps the plant vegetative. Begins to feel the cold, you start now accumulating your red blobs, and that's now switching the gene off. You see the nucleosome, these things compact up, gene is now off. Okay, now spring comes. What colour is going to be my blob at the chromatin at FLC in spring? That side of the room? Green or red? Hands up. Who's going to say green? Who's going to say red? Okay. It's red. It's because it's staying off, it's epigenetically silent, it's important for somehow, in the absence of cold, it stays red. Even though the plant's back in the warm, everything's fine and dandy, it's growing happily, the modifications at that gene stay red. So the gene stays off. So what we've got in our life cycle now is we've got the gene is on, it switches off. It switches off actually because of these polycomb proteins, which involve these proteins I told you about before that came out of the genetics, linked to this polycomb repressive complex 2, this polycomb mechanism. And actually it starts off at one little place in the gene, and then that's what happens in the cold. And then actually at the end of the cold, as the plants grow, that complex, or part of that complex, spreads out and spreads across the whole gene. And we think this is important as a way of being able to switch from one state to another. You've got to sort of, it's like a zipper. Your jacket's wide open, and now what you're trying to do is zip it up. You've got to sort of start and link it up. You know how the zipper at the bottom, you've got to get it fixed? Well, what happens in the cold is a bit like that, because it's quite a difficult switch. But then once you've done that, then you can zipper it up, and actually that gives you the robustness and the stability to stay off through many, many cell divisions as the plant is growing. So that spreading is important. And then the next thing that happens, of course, is in the seed, you have to switch the gene back on again. And actually, remarkably, again, we went in with mutations and said what's important in resetting. And the enzyme that came out was the exact opposite of the polycomb enzyme. It was a demethylase as opposed to a methylase. So, you know, genetics sometimes is very nice. Sometimes it shows you a gene, you have no idea what it is, and you scratch your head and think, what's the mechanism? But for us, it was very nice. It was a demethylase. So that made sense. Now, I'm just going to show you one bit of data, because someone was asking, you know, well, what actually do in the lab yesterday? You know, it's all very well showing these schematic pictures, but what are the experiments? So for the plants and how we look at this, what this does, so here you've got this local site, and now you have a spread site. What you're going to be looking at is the modification. Remember this H3K27 trimethylation. So we take the plants, we grind them up, we take the bits of chromatin, the DNA, the protein, that are together still, and then we add an antibody which recognizes that modification, and we immunoprecipitate. Then we take the immunoprecipitate, extract the DNA, and then do a PCR analysis which just measures how much of the DNA of a very specific sequence is in that immunoprecipitate. So there's a whole lot of tubes and a whole lot of PCR goes on, which is a bit boring, but if you at least have an idea at the end where you're getting to, it keeps everybody going. And then you plot them all out, and you've done the experiment several times, and you can see that actually, so if you look at the black one here, which is just down here, there's not much there before the cold. And then with increasing weeks of cold, you get this increase in this particular modification at this internal site. But it's only when you bring the plants back into the warm that that spreads across the whole gene. Now, if you remember, I showed you it was a quantitative silencing. So in fact, the fact you get quantitative increases in this silencing mark, well, that seemed very satisfying. Like, okay, done, figured that out, done and dusted. Until a colleague of mine who's a computational, who's actually a theoretical physicist by training, of course, looks at this very differently and said, well, but it's a bit strange, Karen. You told me if you give a plant, let's say, 4 weeks cold, it will stay at that level through the rest of the development. How can you partially inherit or inherit a partial amount of a particular modification? Surely in your blobs it's either there or it's not there. Maybe you could imagine it's half there, but you know, this quantitativeness was difficult to explain. And in fact, he was right. We did a lot of analyses, and it's not even in all cells. It's a cell is either green at that locus or red, and it's the proportion of cells that switch from green to red that changes with cold. But of course, which I've represented here by blue and white, but you can see each of these is a cell where the gene is either on, blue, or off. And as you get increasing weeks of cold, more and more and more of the genes, the cells have switched off. But of course, you grind this up, it looks like a beautifully even, nicely quantitative change. But it's not at the cell level. And that's something that actually everybody's beginning to pay attention to now. We call this digital silencing. It's a bit like a computer, you know, you're on or you're off. And in fact, this is something we're going to have to pay attention to, because most assays, of course, we do by PCR. You're averaging over a number of cells. Okay. Sensing. Now, we don't know quite so much about the sensing, but that's, of course, the interesting bit, how the plant's feeling the cold. So we go back to our schematic here, and we've had, this is the polycomb, which is nucleating and then spreading to remember. So the one cold thing I've told you so far is one of these proteins that came out of the screen that was required for the nucleation of the silencing. And when we look at the expression of this, gratifyingly, it increases slowly with the cold. So you think, well, that's really all you need to make this a cold-inducible process. So one question is, how is that regulated by cold? We're doing that in a very intensive series of experiments. Still can't give you the answer, but we're doing that. But the other thing we looked at was what happens at FLC when you don't have that protein. And of course, this is where the genetics helps you. You can do that expression in now a background which doesn't have that protein. And in fact, it was surprisingly normal for the first couple of weeks, the downregulation. It was just, went wrong later and then had no memory. The gene was switched back on again when you couldn't keep the gene silenced. So that made us go in search of what was happening during those early weeks of cold. And that took us into the world of non-coding RNA. Now again, epigenetics is sort of one term, but you'll also hear non-coding RNA as another term that's coming up everywhere. People think of it as transcriptional noise, they just don't quite know what it is, but all these deep sequencing technologies finding more and more and more of the genome is expressed. And so is it functional or not is then the big question. When we looked at these, our non-coding RNAs turned out to be the other strand of FLC. So here's our FLC gene. And what we have is a whole series of things which are changed in their splicing. On the other strand, we have a non-spliced and then two other forms. Now, when we removed them, we had an effect on the ability to respond to the cold. So they did seem to be important. But as I've shown you, what happens in the cold is the cells are switching off in a digital manner. And all of the RNA things I was showing you were sort of, again, averaging cells. So we realized we were going to have to figure out how to look at the RNA in each cell to understand, is the RNA and the antisense digital in the same way? So that's when we came up, and a couple of years ago, we developed this. And we hadn't had it for plants before, but it's really opening up a whole new set of tools for the plant world. And they had developed this with C. elegans, so little worms. And they had made oligos with basically a fluorochrome on the end, so you can see them in the microscope. So the oligos are maybe only 20 nucleotides, so you have lots of them, all hybridizing to the same RNA. You damage your cell just enough to get them in, and then you put your organism under the microscope. And those oligos find every RNA in the cytoplasm. Now what you're looking at here is one plant cell, a root cell as it happens, and a nucleus counterstained with DAPI, which highlights the DNA. And each one of these signals here is a messenger RNA in the cytoplasm of a plant cell. And I think when you can start to see this, then it really comes to life rather than just some graph, okay? So this is how many FLCs are made in a plant in autumn. And in the nucleus, what you're looking at here, green shows you the exonic bit, so this is the proper RNA, the spliced RNA. Red is the actually nascent transcript before it's been spliced. So you can actually look at both forms in the same cell. And if you look carefully, here are the two red dots. One of the red dots is also covered by a green dot, hence it's yellow. These are the two genes in the nucleus where this gene is being transcribed. And you can see it just like that, okay? So this is for the sense strand, because we started with the sense strand because it's much more abundant. So can we see it for this antisense, this important one that I was telling you about, it's important in the cold? Well, it turns out, yes, we can. We can only see the unspliced form. But it was very revealing in that when we looked in root cells now, so this is a slightly lower magnification, so you can see the files of root cells here. So most of the time, no signal in the nucleus at all, because it's not made when the plants are in the warm. Within 2 weeks, almost every nucleus is expressing the antisense. So if you then look at a series of, this is now no cold in the nucleus, this is looking at the antisense, what happens very quickly is that you get these huge, great big blobs being produced. So much bigger than what's happening in the cold, even in these rare, remember these are the rare types that are making antisense in the warm. Great big blobs, which then start to break down into multiple blobs. It's like having a huge, great big jelly which sort of blibs off. And then after about 3 weeks, we're back to sort of the normal here. Now, why I show this, and I showed the X chromosome inactivation, one of the big and interesting features of the switching of the X chromosome that people have looked at in females is that chromosome that forms the heterochromatic bar body is covered by an antisense transcript, and they call them Xist, X-I-S-T. And it's a well-studied, and the chromosome is coated by Xist and stays coated by Xist. And actually, this looks awfully like what's happening at that inactive X. But here we've just got a single gene. And then when you look at both the sense and the antisense on the same nucleus, and so you can do that with different colors, you never see the same gene expressing both sense and antisense. So if you've got one gene, you're either one or you're the other. And so that's a great way, if you switch on the antisense, of shutting off the sense transcription in the cold. So of course part of the sensing mechanism then is the induction of the antisense, and we now need to understand what the cold is doing to this to switch it on. That transiently shuts off transcription. See, this has gone small. That needs to be small for this to happen. So if you transcribe too much, you can't nucleate your polycomb, and then you spread, and then you're stable. And then you switch back on again. Now, I was going to kind of whiz through, I'll whiz through this in very quickly. Lots of natural variation in this process, because winter is different in different places. And in fact, if you do the analysis on the Arabidopsis, you can actually see really very different vernalisation responses. This shows the acceleration of flowering with different weeks of cold. I'm not actually going to go through all of these, because this just shows, in fact, what is different is actually just very subtle things at the gene itself is what adapts you to a winter of different lengths. So that sets us up to do this experiment now to ask, well, why, how can you adapt to different winters, and how are these small changes actually changing the mechanism? So we have some really fun experiments with Swedish colleagues now. We've got field experiments, and these were the sensors that I showed you up in the north. So we have it in the north here near Lurvik, and we have another one in the south here near Ulstorp. And we go in and actually try to understand, when vernalisation is happening and what's the adaptive significance of these different types of FLCs which are giving these different vernalisation responses. So I get to go to northern Sweden, and it's absolutely beautiful up there at the high coast. lots of, and it always seems to be sunny when I go. My lab's very furious because, it's not always very sunny. As you can tell by the master student who then harvests every week through the whole of winter, in the northern Sweden. And here she is, digging 3 feet of snow to get to the plants, to harvest them, to send them back to Norwich. Anyway, this is really the experiment, this is to go back to this question, with this really complex temperature profile, now we've got all the players. When is it all actually happening in a real winter situation? That's what we're trying to understand. So she sent us the samples back. The plants are really very happy, once you've got to them under the snow down here. They don't mind being covered by snow, being this cold. They're really fine. But in fact, this is just a little snapshot of data to show there's some very big surprises coming out of this. So we're doing it in Sweden. We're also doing it in Norwich in a greenhouse where we've taken away all the heating and the lighting, so we can introduce transgenic lines as well. So we can then manipulate each bit of the process and ask, how are those bits changing now, this ability to sense this very complex fluctuating temperature environment? So if you just look at the ones on the left here, here's the temperature profile through I think it was 2014 winter in Norwich. And here is a whole series of different genotypes. And what you're looking at is FLC-RNA levels. So here is sowing in September. And they all look pretty much the same to start with. And then there's a sort of like change. You can see a rate of silencing of a certain rate. And then here, which is really after that first cold spell, the rate of silencing accelerates in the wild type and most of the genotypes. Except these little guys, they seem to have lost the ability to sort of speed up. And those are all your polycomb mutants and those PhD proteins that I was telling you about. So it's really quite exciting that we're now trying to understand, so what's this? This is an early cold phase, and we think this is the antisense regulated phase, and this is now the nucleation of the polycomb phase. But if we can really understand the cold inputs, we can then really understand how the plant constantly is sensing what the temperature is doing day and night through a season, to then be able to predict exactly what might happen as really we are starting to get very, very variable winters, causing havoc with agriculture. How many of you couldn't buy broccoli this year because there just wasn't enough broccoli in the shops in January, We really do have an issue with that. So it's going to be significant disruption. So we really have to understand how plants are doing this to be able to then breed for it. And this is my group. This is actually at the Swedish retreat we go to, where there's this fantastic place where near our field experiments. So you can see the Swedish collaborators here from Lund and from mid-Sweden and the MSc students. This is the group, the 2 people doing the big field experiment are Joe Hepworth and Rea Kurinoti. And this is Martin Howard, who's the theoretical physicist I was telling you about, who we have great group meetings and there's lots of discussions. And he's very influential in terms of, actually integrating temperature as a biophysical type mechanism. So it's very important to have his input. And here are the funders. And this is our strapline from the Royal Society. Diversity makes us stronger. Anyway, thank you.